Welcome everyone to this webinar. Today I will tell you a little bit more about microbiome monitoring. This is basically a service that we at Upwater provide for wastewater treatment plants. And we use molecular diagnostics to optimize wastewater treatment plants. So why do we do that? So the wastewater microbiome is, so to say, the invisible workforce. So here we see a wastewater treatment plant. You see the secondary cleaning, and this is basically biology. So here, basically everything goes in and comes clean out. However, who does this? What we know so far is we know certain bacteria are removing the nutrients and are cleaning the water. But what we at Upwater found out is it is definitely necessary that there is more transparency in the treatment process, because it is crucial to understand how a treatment plant works. We know the treatment plant works like this. You have an inflow, screening, settlement, the biology, and the outflow. Within the system, we can measure multiple things. We can measure the oxygen, we can measure the solid retention time, we can measure the temperature, the pH, certain chemicals, the turbidity, the conductivity, with different sensors, for example, or chemical reactions. And all of that is basically done automatically, and no one questions it anymore. However, we are able also to measure the bacteria and the biology. And these bacteria fulfill a very, very important role within this biology, because what they do is they clean it. So the important functions in a wastewater microbiome are, for example, the carbon degradation, which is done by heterotrophic bacteria, the nitrogen degradation done by ammonium oxidizing bacteria, nitrite oxidizing bacteria, denitrifying bacteria, and for special regions, Anammox bacteria. There is also other bacteria that accumulate phosphate, which are called phosphate accumulating organisms, or glycogen accumulation, which are, so to say, the opponents of the paleo. So if you know them, if you have them, it's maybe not so good if you want to do BioP. Or in anaerobic digestion, the whole concept of anaerobic digestion is based on the degradation of certain carbon compounds to produce methane. And this can only be done by certain bacteria or archaea. Here I show you basically, can you see my mouse like this?
Yes.
Okay, so this is basically just an idea that I can give you of the activated sludge in a real wastewater treatment plant, which is basically the average over the year. And here you see there's 28% of heterotrophic bacteria, 9% the nitrifying bacteria, 1.5% AOBs, 4% NOBs, 3% PAOs, and 1% GAUs. And 47% are actually undefined in terms of their function. This is where research comes into place, because this is their job to find out what are these bacteria capable of. What we are doing is basically we can assign the bacteria that we find now to their specific role and can say, okay, this is the amount of bacteria you have in there. Here, for example, we have a yearly campaign over the whole year. Again, we have the heterotrophic bacteria, the PAOs, the NOBs, the AOBs, and the denitrifying bacteria. And you see, there's quite some dynamics going on over the years. So we start here in January and over the whole year. This wastewater microbiome, it is important to understand that the stability is the key. If you have a stable wastewater microbiome, everything will work smoothly in your wastewater treatment plant. However, it's like domino. If one of the stones falls out, it doesn't work anymore really well, because this wastewater microbiome responds to the composition of the wastewater. So that means toxic compounds or other bacteria that come within it, the operational conditions, poor sludge control, inadequate aeration, the sludge age, environmental factors such as precipitation, and the seasonal changes. All of these factors can harm the wastewater microbiome, which means you basically get a problem. And then you cannot fulfill, for example, the treatment process. Therefore, your water gets not cleaned very well, Or this can also include insufficient nutrient removal, bulking sludge, foaming, and other problems. Causes of certain process instabilities within the wastewater treatment plant can only be explained with DNA measurements, because very often it's the case that you just basically lose certain bacteria and you cannot do the function is not fulfilled anymore. At Upwater, we have developed a service called microbiome monitoring, and we offer DNA sequencing instead of laborious microscopy, which is quantitative, objective, and cheap. We provide solutions of operating problems to improve the effluent quality and the targeted use of chemicals, the cost reduction. I will give to all of these later on some examples. The way it works is like the wastewater treatment plants send us samples on a weekly basis, on a monthly basis, or just like randomly during the year. They send it to our lab. We analyze the DNA, which means like we sequence the complete microbiome within this sample. So we have knowledge about every single bacterium that is in this sludge at this certain moment of sampling. And we do data analysis and data delivery this within 7 days, or even possible within 4 days. And this allows then the wastewater treatment operators to really use this data to treat their water, or influence their process, the way they operate the treatment plant. So where can microbiome monitoring help? So one thing I told you at the beginning is this transparency. It's like, I know a lot of wastewater treatment operators that tell me like, yeah, okay, I know my plant very well. And then if you ask them like, okay, how does it look in microbiology? Of course, they say like, we don't know. So I think know your plant is a very important thing for every wastewater treatment operator. And we are still trying to convince a lot of them. And here we go into the biology and aerobic digestion. So here we see, this is a campaign. This is a wastewater treatment plant in Zug, in Canton Zug, here in Switzerland, that we actually accompany since now nearly 3 years. And these are the top 25 bacteria and their abundance over the year. Here's the time, and here is the average abundance, the abundance. And each of these colors, of course, represents a different genus of a bacterium. And what you see is like, it's very interesting. We actually have similar patterns every year. And this is what I mean with know your plants. You have seasonal changes of the microbiome, but they reoccur every year. So if you have knowledge about it and certain seasonal occurring problems are repeating each other every year, you can beforehand know what happened, what will happen, and take active measurements against it. Another application that is very important and is basically one of our best selling points is like basically who was responsible for settling issues, for foaming, or very complex operational problems. Here I also have some good examples. For example, for a seasonal nitrite accumulation. Nitrite is a toxic compound, especially for fishes. And when it gets released in the environment, it causes big problems. Here we see another time series of NOBs over time. So here we have a genus, a very common genus that does the nitrite oxidation called Nitrospira in black. And on the first axis, you have the occurrence in percent, the time, and here you have the nitrite. And this is another genus of bacteria, of NOBs, which is called Nitrotoga. And as you see, they don't co-occur very well together. So either this one is dominant or this one is dominant. This one is dominant, or this one is dominant. This might be not such a big deal. However, in green here, you have nitrite accumulations. Every time we see a shift between these dominating species or genera, we see high loads of nitrite that accumulate until the next genus basically takes over. This is a big problem because in Switzerland, we have certain rules of how much nitrite you can actually release. And if you go above 0.3, you get a problem. And this is something wastewater treatment operators don't want to have. So with our analysis, we could actually figure out, okay, these changes actually can explain why nitrite was accumulating. And we are strongly working now also on reasons to mitigate these nitrite accumulations. Operational monitoring. There's certain operating modes like moving bed bioreactors, granulation process, or the Anamox process that are quite sensitive and not that easy to operate. So our services actually help to understand the system better and actually help also to run the system better. For example, I have here a moving biofilm reactor. And one big issue, especially in wastewater treatment plants all over, is the winter. Winter means lower temperatures. Bacteria don't behave as they should be. They are much slower. You really have to adapt your wastewater treatment plant to the situation. In a moving biofilm reactor, you have two compartments. You have bacteria on the carrier and you have bacteria in the sludge. The idea is you accumulate most of the bacteria on the carrier because they're safely in a biofilm over the winter and can perform quite well. In this problem here, you see we have the carrier and the sludge. One problem is that if you don't do DNA analysis, you don't really know where the bacteria are. Okay, I see nitrification works, but who does it? Is it the carrier or is it the sludge? So this is the first thing that we actually can inform the operators. Where are your bacteria? In this problem, we had the operator told us, well, my nitrification doesn't work. So we did weekly DNA analysis for 4 weeks to just check the status quo. What we found was there are quite some NOBs here, so bacteria that use the nitrite, but there's hardly any AOBs that can use up the ammonia. So nitrification might not work that well. So we have to think about, okay, what do we do? What can the operators do? And then, of course, also monitor the success. So what we did was we increased the sludge retention time. So sludge is longer in there, bacteria can grow better. We told them to increase the oxygen demand and also to return sludge, to increase the return sludge ratio, to have more nitrite in the biology. And what happened was that within this year, we managed actually to make this system completely winter fit with huge amounts of NOBs on the carrier, together with also an increasing number of AOBs now, and lots of AOBs in the sludge, that when certain certain seasonal changes are starting, they can actually move also on the carrier. Here, this is a special case because they also have a lot of Comamox bacteria, but I will not go in detail to that. They also take the ammonium up. So the results is that we had significant increases in AOBs, NOBs after the recommendations, and the nitrification was fit for the winter. Another very sensitive process is the granulation process, such as Nereida or using a hydrocyclone. Here we can provide quality assurance and success monitoring. For example, here we have filamentous bacteria in the granule and in the reference. Filamentous bacteria are good to stick the sludge, to hold a sludge flock together. However, if you have too many, it also can cause sludge bulking. Granules are actually formed to avoid this. And here you can actually see that filamentous bacteria grew like crazy in the reference, but in the granules, they stayed the same. And the anobes, the ones that you want, stayed quite similar in the reference, but grew very, very strongly in the granule. So the microbial monitoring of the granulation process informs you about, is it going in the right direction? And also gives you the early detection of positive and negative bacteria. The same in an Anamox startup. Anamox are also very sensitive bacteria, and especially if NOBs are around. Here we have Anamox bacteria in red, the AOBs, and in green, the NOBs. If you have a lot of NOBs, they take up the nitrite, and the Anamox doesn't have enough food to actually perform. So what we did again is like to check the system, figure out, okay, this is the problem. Then we implement the measures, reduce DO, and additionally added sludge liquid, so there was enough more food available. And what we managed was like, okay, we get less NOBs, the Anamox could grow in, and the Anamox reactor really worked well now. Another important thing as an application is the early warning, such as growth of filamentous bacteria, unwanted polyaccumulating bacteria, phosphate accumulating bacteria, and the NOB growth in Anamox, as I saw you before. Here, I show you the filamentous bacteria and the sludge volume index. Sludge volume index is basically an idea of like how fast the sludge settles. Here, we have a certain bacteria that is called Candidatus microtrix. This is like one of the strongest bacteria that cause sludge bulking, together with another bacterium called Leptotrix. And this is a monitoring we did at the biggest wastewater treatment plant in Switzerland, which is Wertherzli. And if you look at the blue line here, this is the sludge volumes index. Every time it's high, the sludge settles really badly. That means you need much more time to clear the sludge than if it's low. And you see every time this bacterium grows in, afterwards, the sludge index goes up. And this is quite easy to target. So with our monitoring approach, it actually allows us to tell the operators, look, microtrix is growing in, use aluminium, which is the common strategy to get rid of this bacterium, to get it out. Here, the problem is that most of the wastewater treatment plants don't really look what bacteria they have. So here we have another wastewater treatment plant. We have the SVE, the sludge volumes index, but also the microtrix. And we see microtrix is not here anymore in red. It's other bacteria. The problem now is when adding aluminium here, it actually affects very well the microtrix. However, at the end, the second half of the year, there was no microtrix. They still had problems with the sludge volume index. However, this aluminium did not help at all. So they spent around 25,000 francs on aluminium that was not necessary. Here is also the issue that not all microtrix is the same. And only with a DNA analysis, you actually know which species of microtrix is present because they react differently to aluminium. So overall, in this sense, it makes really sense to do the DNA analysis, especially when you have problems with filamentous bacteria. And last but not least is the quality assurance or the success monitoring. Here, testing new mode of operation, coagulant, or other changes allows you to understand how does it work, does it do the job, or can you completely forget it? So, as a wrap up for the microbiome monitoring wastewater treatment plants is that the invisible workforce made visible. The wastewater microbiome drives crucial treatment processes, and our services provide unprecedented insights in this complex community beyond traditional monitoring. Understanding is the key to stability because it's very dynamic and sensitive to operational and environmental changes. the monitoring can help the diagnosis of the root of instability, for example. We have diverse applications for optimization, such as troubleshooting, process control, early warning, and targeted actions. And it leads to improved effluent quality, targeted interventions, and potential cost savings when using, for example, less coagulant. And as a conclusion, microbial monitoring is a powerful diagnostic tool for modern wastewater treatment, offering deeper insights and enabling more precise data-driven management of biological processes. So far, we have 30 wastewater treatment plants in Switzerland that use our services, with 5 wastewater treatment plants now in Germany, as well as 5 wastewater treatment plants in Austria. And we have strategic partnerships with different coagulant distributors that also send us on a regular basis samples. And this is our team with Jonas, Wenzel, Robert, me, and Manuel. We are the management of the Upwater, and Karen is my lab technician, and Adi and Remo are our project managers. Thank you very much.